Many geneticists work with Drosophila. They're small, they're easy to keep, and because you can work with large numbers, you can study rare genetic events, like recombination between genes, for example, or rarely mutation. Some geneticists work with bacteria because all of these are true to a greater degree. They're even smaller. They're even easier to keep. Generation time is much shorter. And it allows you to study rare events, such as new mutations or comparing mutation rates, given that overnight you can create millions and millions of new bacteria. Some geneticists work with virus or phage for the same reason. They are even smaller. They can be stored indefinitely given that they are not alive. And a milliliter may contain millions and millions of bacteria, but it could contain billions of virus. A favorite to work with are those viruses which only infect bacteria, known as bacteriophage. And there are different strains of both the bacteria and the virus. Some strains of bacteria are more easily infected. So for example, in these examples, E. coli strain K12 is a restrictive host. Only wild-type virus will infect it, while E. coli strain B is a permissive host. Both the wild-type virus and the mutant virus can infect it. As indicated, there can then be mutations and thus different strains of virus, with some having different virulent properties than others. One can then study rare genetic events, such as a mutation which might convert a mutant strain, which has a point mutation, back to the wild type strain. And so thus one could study at what rate would one specific nucleotide, just that one spot, mutate to revert to the wild type condition. Note that only some mutations would allow this. So for example, a deletion mutation would not allow this. So a point mutation would not convert this deletion mutant back to the wild type strain. One could study another rare genetic event, recombination within a single gene. If one co-infected the same bacteria with two different strains of virus, recombination might occur so that some of the recombinant viral DNA nucleotides within the infected cell would contain both wild type versions because recombination had occurred between a mutant which had a normal portion of the first region of, say, a gene, and a different mutant which had a normal portion of a second region of a gene. Now, this would not always work if there was a mutant with a deleted region, or if two mutants had mutations in the same region of the gene or even at the same nucleotide. But this would be an example of an extremely rare genetic event which could be studied using virus because one can work with such enormous quantity of virus.